thank you all for coming out for this week's colloquium. I think many of you that work in the building will understand when I say I'm critically relieved there are no bats in attendance, at least not in person today. So that's a step up from yesterday. So I'm glad to have all of our human attendees. And we're really looking forward to hearing one of our late summer seminars, which I believe is going to be kicked off by Rebecca. So I'll turn it over.
All right, well, introduce Rebecca. So it's my pleasure to introduce Rebecca Salamon. She's a graduate student in my lab and study heart regeneration, but really study how cells control also heart function. And today, Rebecca will be talking about her thesis project, which really had some very cool findings and even
hey, Tim.
See it. So I'm looking forward to Rebecca's talk today. So Rebecca, take it away.
I'm just gonna mute this really quick. I think my side is muted, so hopefully that was just a glitch.
Yeah, was that from?
It kind of sounded like it was from my computer, but I have my thing muted as much as I'm not really sure. So just if you're in attendance, don't talk virtually. It sounded like it was from the virtual people. So just make sure you're muted if you're on Zoom, and then we should be good. Cool. Well, I'm really excited to be here and present my thesis work. And today I'm just going to be talking about the developmental portion of my work. Like Ahmed said, we study a lot of heart regeneration, but one of my favorite parts of my project has to do with heart development. And that's kind of what got us kicked off on this project. I do. Yeah. Thank you. So it probably doesn't come as a surprise to most of you that the heart is the first organ to develop during embryonic development. And just 22 days into development, the heart will first contract, leading to the embryo's first heartbeat. And I think if we take a moment to appreciate the heartbeat, it's pretty remarkable that it starts at embryonic day 22 and lasts throughout our entire life. And it does so rhythmically, and it's very dependable. So how does this cardiac signal actually work in the heart? That's mainly due to the cardiac nodes. The nodal cells are these groups of specialized cells that allow the heart to beat, and they're able to do it reliably because they can undergo a process called spontaneous depolarization, which means they don't need any signals to contract. Essentially, they are their own signal, and that's why you have such a reliable heartbeat. But what I'd like you to appreciate most from this figure is where the nodal cells are located. We have the SA node and the AV node, and they're both within the atria of the heart. The atria are these top two circular regions, and they're not in the ventricles of the heart, these bottom elongated regions. They're in the atria specifically is because they're able to propagate the signals. They don't need to be everywhere in the heart. They pass them along through His bundles in the atria and through Purkinje fibers in the ventricles. And so, of course, your heart rate is not always going at the same beat or same rate. It's increasing or decreasing depending on external stimuli, and it's able to do that thanks to the autonomic nervous system. And that can be divided into two main branches: the sympathetic or the fight or flight nerves and the parasympathetic or the rest or digest nerves. They're nicknamed these appropriately as the sympathetic nerves increase heart rate and contractility, whereas the parasympathetic nerves decrease heart rate and contractility. Most interesting to me is where this innervation is thought to localize. Both of the parasympathetic and the sympathetic nerves are known to localize at these nodal cells. They play like a game of telephone, sending the signals to the nodal cells if they need to increase or decrease heart rate, and then the nodal cells propagate the signal. So thus, for controlling heart rate, there's no need for them to directly innervate anywhere else. And so standing in the field is this model where the parasympathetic and sympathetic nerves are located within the atria, but unique to the sympathetic nerves, they're able to directly innervate the myocardium, the ventricular myocardium, whereas the parasympathetic nerves are thought to be completely void from the ventricles of the heart. So why are the sympathetic nerves in the ventricles of the heart then? They are there in order to regulate vasoconstriction and vasodilation of the coronary vasculature. And they're able to do this by directly innervating the arteries and veins of the heart. And we see here again that the parasympathetic nerves are not thought to do this function. It's supposed to just be the sympathetic nerves, and the parasympathetic nerves are once again just within the atria of the heart. And so it's been recently challenged that not only do the nerves control heart rate, and not only do the nerves control vasoconstriction, vasodilation, but that there are many more nerves in the ventricles of the heart, that their function must outstretch just these two simple models. And so a few new papers published just within the last year have highlighted the role of the sympathetic nerves functioning outside of just these two boxes. And these all have to do really with them functioning with the main cell type of the heart, the cardiomyocyte. So we see here in the first publication, just later in 2021, where they were able to show that the sympathetic nerves are able to regulate cardiomyocyte proliferation during development. It was also shown that the sympathetic nerves can regulate cardiomyocyte size, whereas nerves that directly innervate the cardiomyocytes lead to bigger cardiomyocytes, whereas the cardiomyocytes that are not innervated are smaller. Also, we see that this interaction between cardiomyocytes and nerves are absolutely essential for the nerve survival, where the cardiomyocytes feed neuronal growth factor to the nerves, which allow for cell viability. And so we can see through these findings, I think readily apparently, is that they're all focused on the sympathetic nervous system. There's no real data here about the parasympathetic nerves, which led us to ask the question, well, how do the parasympathetic nerves pattern and distribute in the heart? And by understanding the patterning and distribution, we can start getting indicators of how they're influencing cardiac physiology. And I think the importance of this is really highlighted well by a quote by John Coote here. And he's a professor of physiology at the University of Birmingham. And he's been a huge advocate in the field for us exploring the role of parasympathetic nerves, especially in the ventricles of the heart, as there is sparse data out there showing that the parasympathetic nerves must be there and it must be influencing cardiac physiology. And it's very important that we understand the patterning and physiology of this, as it will lead us to better understand heart dysfunction. And I'd like to add to this quote, not just dysfunction, but also for us to better understand heart development and heart homeostasis. And so it's with these questions that led into the branch of my project that I'll be talking about today, which focuses on exploring the 3D patterning of the parasympathetic nerves during development in the cardiac ventricles. And so, of course, our first question was simply, are the parasympathetic nerves there? And in order for us to answer this question, we had to come up with a new method. So if we would ask the question, what do the parasympathetic nerves or what do the nerve axons in the heart look like over the years? This timeline really shows where the field was at and what the field was able to identify. And so we can see in the 1960s, you're really limited to bright field microscopy, electron microscopy. You're not really able to gain much data. You're only able to see very large nerve structures such as somas. In 2010, we started to see some data arise, two-dimensional data, histological sections were being taken and stained with nerve markers, and we start to see a few nerve axons being identified. But if we can take a moment to imagine working with 2D data and trying to make a 3D model, when you're sectioning the heart, you're damaging the nerves. And when you're reconstructing the nerves, it's also very inaccurate. And so that's why in 2014, the field progressed to whole mount imaging. Whole mount keeps the tissue intact, so you can look at the heart intact. This is extremely important for things like nerve fibers that are so fragile and thin that you can't accurately evaluate their morphology in 2D. And so progressing into a 3D model allowed for some new visualizations of the nerves. It also allowed us to see that there are many more nerves than previously thought. But what I think you can appreciate from both of these images is the fact that early on, it was very inaccurate. So in this image, we see that the red is inference data due to high background. And then in the 2017 image, we can see that there's still high background. That's very hard to tell, very high signal to noise ratio. And so what we did was try to take these techniques where we were at that involved not only whole mount microscopy, but also tissue clearing. Tissue clearing is going to remove some of that background, also allow us to go deeper into the tissue and visualize structures that are deep within the heart. And we developed a tissue clearing method, a whole mount staining method, and a confocal imaging method, all compatible with the heart being entirely intact. And so we started off with an uncleared heart. We send it through passive clarity. What that is essentially putting the heart into heavy detergents and heat. And those detergents remove the pigment from the heart, which is mainly in the form of blood. Once that is all removed, we can stick it into a refractive index matching solution called RIMS that homogenizes the particles, allowing light to evenly pass through the tissue. And so then the tissue is completely transparent by the end, allowing us to get really high resolution images and see what is really intact, what the nerves look like in the intact heart. Very important to my studies was that it was also compatible with immunostaining. Many of these new clearing techniques are not compatible with immunostaining. They cannot bind antibodies efficiently. However, mine being a more gentle type, using more gentle type reagents is able to be compatible with immunostaining. So we can add in markers for our cell types of interest. And then we go to image using a 3D printed well. We stick the heart into the top of this well. It sits at the bottom of the well. And then we can image using an upright microscope. And lastly, to get the most high resolution images, we use pick stitching. And so each panel or tile is taken at 4x, or I can go up to 10x zoom and then stitch together. This allows me to get a high resolution image, but then keep it where I can see the entire heart in one image. And what I hope you can appreciate is the fact that we have been able to see more nerve fibers and analyze these in 3D like never before. And so this allows us to start getting a 3D scope into what the nerves look like in the heart, which really has not been rigorously analyzed in the field. And so since our cell type of interest is the parasympathetic nerves, we went with a reporter model. The reporters allow for high fluorescence, which work really, really nicely with the whole mount staining and imaging techniques. So in order to generate my model, we're using a choline acetyltransferase TD tomato mouse. So we crossed the reporter mouse, the Rosa lock stop locks TD tomato mouse, to a Cree driven under the endogenous chat promoter. So in all the cells expressing Cree will excise the stop codon, leading to constitutive expression of our TD tomato reporter protein solely in the parasympathetic nerves. And so to begin, we just took a mature mouse heart at postnatal day 7. I'll abbreviate postnatal days as P and embryonic days as E throughout this. Underwent heart clearing, whole mount staining and imaging, looking for the TD tomato protein. And at the end, a very important step is the fact that I don't do the analysis. I pass off my hearts now to some great collaborators. We have a great undergrad in the lab, Corva, and a graduate student, William, who are running analysis so that way we can see what our models look like in 3D using Amaris. And so we were very surprised to see the first look under the scope that there are dense parasympathetic nerves in the ventricles of the heart, many more than previously described or ever described, really, and that they cover nearly the entire surface of the heart. And they're not there in small quantities either. We see that there are dense nerve bundles and really nice nerve axons shown throughout the heart, suggesting that they play a much bigger role in influencing cardiac physiology than seen before. So of course, we were wondering how these parasympathetic nerves, that patterning is compared to sympathetic nerve patterning. And so in order to look at sympathetic nerves, we stained for the sympathetic nerve marker tyrosine hydroxylase. And what we were able to see is that the two patternings reflect each other nearly perfectly in every single way. And when we overlay the image, we can see really nicely that these nerve fibers or these nerve axons are densely intertwined, not only in these dense axon bundles, but also even in these smaller regions. They're perfectly aligned, suggesting there's a cool cell-cell dynamic there that we're probably not going to get into too much. And then if we go ahead and look at this bundle and we bisect it, or we look down the tube of the axon, we can see nicely that they're bundled together. And that's a phenotype that I've been able to see across all the regions of the heart. And so we were able to see that the parasympathetic and the sympathetic nerves are densely intertwined. And so here I've shown you a max intensity projection of all of the Z stacks that are taken when I'm taking images on the confocal. But since it's taken as a Z-stack, that means there's quantifiable data about its 3D architecture as well. And so in order to look into that, that's where we are using Amaris. I can upload all of my 3D data into Amaris and reconstruct this in order to get information about the nerve spatial location, of course, but also its density and its volume. And we can start quantifying how the nerves are innervating the heart, what depths are they innervating, where do they differ between the sympathetic and parasympathetic. Right now, we're seeing no differences between the two. And I would also like to mention that I do have, of course, two sides of the heart, but for simplicity's sake, I am usually just showing one side. And so what that 3D reconstruction looks like when I take these nerve fibers is, you can see really nicely, it accurately mimics what I'm seeing under the scope. And this is where we can start quantifying some of these really cool phenotypes, such as branching degree and density. My next question I asked was, how are these parasympathetic nerves patterned? Since we see that they're densely intertwined with the sympathetic and parasympathetic nerves, I wondered if they were patterned together or during two separate phases of development. And so in order to look at this, I harvested hearts at embryonic time points that were known for when the nerves first reached the heart through a single postnatal time point. The hearts went through a similar pipeline like I've already described, with the addition, of course, of the sympathetic nerve marker, tyrosine hydroxylase, and again was analyzed with Amaris. And so what we were able to see at embryonic day 15.5 is these nerve fibers just begin to reach into the ventricles of the heart, and they're very, very small and thin. By 16.5, these nerve axons have reached, some of them have reached the apex of the heart, and by E17.5, these nerve axons are very dense and distributed throughout the entire ventricles of the heart. And if we take a look at the rotated Z-stacks here, looking down the bundles of the nerves, you see they nicely progress in size. And each nerve bundle I'll mention that I'm looking at here is considered a primary axon. So it's one of the more dense axons, and they're more dense because they're the founder axons of the heart that initially arise during development. And so they're the ones that are going to first reach the apex of the heart, and they're going to have more time to mature. And that's very important because that's how we are going to quantify things in Amaris, such as branching degree. And so we're able to highlight a primary axon and ask how many secondary, tertiary, quaternary branches are coming off of each of these. And when you start categorizing patterns like this, this is where Amaris becomes extremely helpful, because patterns we might not see by eye become readily apparent in Amaris. And so if we highlight just one main branch, one main primary axon, and its corresponding branching, we can very easily see that E15.5, we've got a branch level of around 2, 16.5, we're increasing to 3, 17.54, and then by P7 in the mature heart, we're having a branching degree of up to 18. And so it's really replicable across my samples as well. And so we're working on quantifying that right now. And this is really wonderful work, again, done by an undergraduate in the lab for Halby. And lastly, to get a more physiological aspect to my project, we're wondering how the nerve patternings or context are patterned in the context of the cardiovascular system. And in order to look into this, I'm using a new reporter model, a connexin 40 mouse. These connexin 40 cells, or connexin 40 is a specific marker of arterial cells. So they are expressed in all of the arteries of the heart, as well as in the pacemakers of the heart. So we'll see the atria light up there, which isn't a problem because I'm only focused on the ventricles of the heart. And they'll undergo a similar pipeline, but I'll highlight the main difference is I will use a pan-neuronal stain, since we've already looked at the differences between parasympathetic and sympathetic nerves. And so what I'm really looking to see here is how do the nerves pattern in relevance to a structure that we know they form a structure-function relationship with. And this has really never been analyzed in 3D, so no one has ever rigorously looked at how the neurovascular patterning is developed and maintained in a 3D context. And so this is really new and exciting, and I've been really looking forward to further doing this data. And so what we have been able to see in the mature heart is that TD tomato lighting up all of the arteries here really nice job of seeing the coronary arteries. If we overlay this with the pan-neuronal stain, beta tubulin, we can see that the nerves and the arteries are very close to each other in proximity, that they pattern similarly, not perfectly. And the fact that they don't pattern perfectly, but there are many regions that we can see that they're in very close proximity, tells me two things. First of all, of course, there's interactions between the nerves and the arteries. But second of all, there's also other interactions that these nerves have, right? And that's something that I highlighted in the intro as well, that we're seeing that these nerves are also forming other cell-cell interactions with not just the arteries, but also cell types like the cardiomyocytes. And really interestingly to me is when I was under the scope looking at how like two of these, this main nerve bundle and this arteries pattern closely together, I was able to see a really cool structure, which is a main nerve fiber being next to one of the main arteries, and then having these axonal projections that then wrap around the arteries. And this goes to highlight why we appreciate these this data being all in 3D, is because we can start to see interactions that we might not be capturing in 2D. And so we can see here the nerves and the arteries interacting really closely, and that is indicative of why the nerves are able to influence things like vasodilation and vasoconstriction. And I'll just briefly go into rigor and reproducibility. And I want to highlight what I'm doing in an imaging-based project, since you all have heard a lot about mice work up until this point. So first of all, one of the most important things that I do is I log every sample that I get online, and I use this through Lab Archives, a digital lab notebook. And this allows me to ensure that nothing could ever happen to my data since it's digitalized, and also keep really good notes over what stainings and what time points these hearts were collected and things like that. And that's great because if I get my hearts under the scope, something doesn't look right, I can always go back and see if there was anything that might have happened during the process, and also just reaffirm that the stainings that I think they got are truly what they did get. Second of all, I store everything in two places, and on an external hard drive as well as a lab server. And I try to store them in the same manner, so that way I can easily find my images, but I also can easily pass off my images with collaborators. It's very important that I can organize my images, find my images quickly, and that nothing happens to them, which is why I back them up to two places. And third, one of the easiest ways I found to find the images that I need, if I'm going to use them for a presentation or I want to go back and edit the photos again or reanalyze them, is I have a giant Keynote file that has every single image that I've ever taken. And so I can just scroll through it, find the heart that I'm thinking of, and then see where I was able to save it, rather than trying to open up a million documents and find the heart that I might be looking for. Since after a while, all of the hearts just blend together and it's really hard to remember what I did, 7, 12, 21, I'm not going to remember that. Yeah. And so with that, I would like to thank my lab, Ahmed, for his great mentorship, Horva Halbe. She's been an undergraduate in the lab, BME major, and she has done a great job of becoming our Mars Pro, as well as William Kasberg, a graduate student in John Audio's lab, and he's been amazing in heading the Amaris side as well and creating a 3D model for all of my data. And so with that, I'll take any questions. Yeah. That is where I'm at right now. I tried snaptophysin, but it didn't work. The first marker I looked for didn't work. And that's just a whole mount imaging or staining issue. A lot of antibodies don't work with whole mount staining. So it's just a matter of me finding a good one. I was going to try synapsin 1 next, but yes. Yeah.
Models of heart disease or heart attack or exercise.
Great question. That's the perfect question, really. We do. And the other half of my thesis work focuses on looking at the heart during not just development, but during disease and regeneration. And this is going to be my favorite. I couldn't present that today, but we were able to, let's do this. We're able to see in the diseased heart that after injury, if we injure a heart that is not able to repair itself, that in the diseased region, there's no nerves. So if we look at it at the region close to, we see that there's a pathological innervation. I showed you that all the nerves are always intertwined during development, but there's pathology in the fact that the sympathetic nerves are able to hyperinnervate in these border zones without the parasympathetic nerves there. And it's the first time I've ever been able to see an uncoupling of the two nerves. And if we look in the infarct zone, there's absolutely no nerves there. It's completely denervated. And if we injure a mouse that can regenerate its heart, a newborn mouse, we see that in the same border zone, there are dense innervation, and that phenotype that we see in the normal heart of the two nerve types being intertwined is there. And then in the infarct zone, remarkably, there's restoration of this innervation. So this is the first evidence of physiological reinnervation of the heart, and that these two nerve fibers are coupled again in perfect coupling like we see during development. And so we were very excited to see this, and this is the second half of my project. Yes. Yes. 100%. And so that is the other part of this as well, is we want to look in the neurovascular remodeling. The coronary arteries are able to do this really cool thing where the right coronary artery and the left coronary artery are usually separated, but during regeneration, they branch. So that way the right coronary artery that was not injured can supply blood to the left coronary artery that did get injured and has a blockade, and it rescues, instead of having to remodel the entire arterial network, it rescues basically those arteries that might die without blood flow. And yes, so we're very excited about that. Any other questions? There might be a chat question. Yay. Cool. Ben.
So really, really cool stuff. Rebecca, great job. I was wondering, the tissue clearing is like pretty amazing to me. Do you know if people are using it in other organs or other?
All the organs. They have taken a whole mouse and cleared it. So yeah, all the organs, and it is so powerful, and just not having to look at 2D data anymore has led to so many insights about architecture that you just would never see. Yeah, they have. If you ever want to clear something, you just let me know. Francisco, do you have a question? Yes, Rainbow is not for me, but another graduate student. We had Rainbow in the lab, and the limitation we faced was there weren't the lasers here to image it, because we had red and orange together. And since the wavelengths are so comparable, we couldn't separate them out. So our limitation was imaging, but we got them set up and it worked 100%. And that is another project that's going to happen in the lab we have a collaborator at UCLA that has the Rainbow mice and has the imaging facilities needed. And so that is next in this project, and I'm very excited about. The Rainbow model is a clonal expansion model, for those of you that don't know, and it's something that we looked into to see how the nerves develop in terms of expansion and division and patterning. Yeah. So, great. Well, I'll let Khagani go because I know I'm a little bit over. Okay, you're set up. This is
Okay, there we go.
computer, and so Khagani is a PhD student, and he's jointly advised by me and UW-Madison. So our lab focuses on our lab of computational biology lab. We develop computational methods to look at problems in gene regulation and gene regulatory networks. And in particular, we are very interested in looking at questions about how regulatory networks determine different types of cell states in cell state. And Rupa's lab focuses on cellular reprogramming. And so we always have wanted to figure out ways to define gene regulatory networks and then what networks actually make different cell types to program and what control efficiency. But we really needed somebody who is willing to work at the interface of computational method development and also really carry out experiments. And then we were fortunate to meet somebody like Khagani. So Khagani is he gets to have two advisors, two lab meetings, two of everything. And now he's willing to become to work at the interface of computational and biology. And so very excited to have him in the lab and he's a lot about
Thank you so much, Sushmita. And after seeing nice wet lab data from Rebecca, time to switch to some computational data. So I'll be talking about the inferring gene regulatory networks that controls acquisition and maintenance of pluripotency. This is a big title, but I'll be focusing on the maintenance of pluripotency today. So I'll be telling you about some background and showing you some data, and of course conclude with the future directions. So during embryonal development, after zygote formation, they give rise to blastocytes, which contains inner cell mass, as you see as a blue, which are pluripotent cells, and they give rise to different germ layers, and they give rise different differentiated final cells, where they became unipotent. So we tell, as cells go through this developmental process, they lose their pluripotency state and became final committed unipotent cells. So we can obtain pluripotent cells from the model organism and then harvest them, and then maintain them in the lab. And while we are maintaining them, they can still keep their two important features. One is self-renewal, and one is being able to differentiate any cell line. So speaking of self-renewal, it means they are keeping their pluripotency state. And we are interested in studying what's the transcription regulation of this keeping their current state. So when we say transcriptional regulation, in simplest form, we know like there are transcription factors controlling other genes. In this example, we say X1 and X2 are controlling the expression of the gene Y. And as they form a kind of network. In the simplest form, we say X1 and X2 are regulators and Y is a target, and this makes a gene regulatory network. So what we are seeing here is already known established core net part of the gene regulatory networks that helps to maintain the pluripotency. As you can see, there are like a major players of the pluripotency like NANOG, OCT4, and SOX2, and how they are interconnected and how they are like supporting each other with feed-forward loop. Just trying to say it's so complex actually, and then it's hard to decipher all different components one by one. So how would you do that? How would you identify this network? There are experimental approaches you can apply. For example, you can knock down overexpress or knock out certain genes and look at the downstream effect. Or you can use a chip, look at the direct binding of the transcription factors with certain parts. But there are like some limitations. For example, if you know like this wet lab stuff are kind of time consuming, expensive. And sometimes it might not be it might be binding, but might not be regulating. So it's informative. It's also limited by antibody availability. Another big problem is it is hard to apply this wet lab stuff in a genome-wide networks. Okay, so that's where the computational approach come to help. So people have been developing different algorithms to model the relationship between regulator and the target using the expression data set. So it's simply based on the idea that if there is a correlation, if one gene is controlling the other gene, there should be correlation between their expression levels. And different algorithms try to use different mathematical and probabilistic approach to model this relationship, right? So one we are using, which is developed by a Roy lab called Merlin PTFA, relies on the same idea. It is originally designed as a Merlin, which was using the RNA-seq data set, doing the looking at the relationship between target and regulator and then inferring the network. And it later evolved to Merlin P and then later Merlin PTFA, where P stands for prior and then TFA stands for transcriptional factor activity. What does that mean? It means while doing the network inference, it considers the already established network not just only consider is apply network components analysis algorithm to identify the transcription factor activity, simply saying identify if already known network is relevant in our context or not, and then use this information to do the network inference and then come up with a final network like this in SSRB. SSRB is like a major regulator here, and you can see its targets is in the red boxes. So we have like a algorithm we can network inference. So we did the experimental design. So asking again the question, what network helps the stem cells to keep their state, right? So the experimental plan was like this. So the Ali, the postdoc from the like former postdoc from the Roy Lab, applied this merely PTFA approach to all the published data and established predicted network. And actually, Caleb and Sunny Grace from different labs worked together and did the low throughput validation successfully. However, we are interested in more like a high scale validation because networks are big, right? And we want to refine more and more components of it. So what we came up with, what I followed up is, so basically take the prioritized list of the identified predicted regulators and do the pool high throughput knockout and subject cells to single cell RNA sequencing to identify, to validate this network in a high throughput way. So the method we are using here, it's called perturb-seq. This is simply combination of CRISPR-Cas9 based knockout with single cell RNA sequencing. So how it works, we basically design different plasmids with different guide RNAs, each aiming to knock down different regulators and pull them together and then do the transduction and then subject cell to the sequencing. But the key question here is, how do you know which cell has got the which guide RNA, right? Because we are doing in a pool fashion. There's the important features of plasmid here we have to see. Of course, they have to contain a guide RNA site. They have a promising select, like a promising and BFP as a selection marker. And the most important feature, each plasmid here contains unique barcode, which we call the guide barcode, which they label indirectly guide RNA. And another important feature is, as we know that from single cell sequencing data, they rely on the poly A tail capture. So basically, a single cell only gives us about the messenger RNA. So this guide barcode is particularly located just before poly A tail downstream of BFP. So it's going to be captured by single cell RNA sequencing, and it will tell us the identity, perturbation identity of the cell, as we know already this relationship beforehand. Okay, so we designed the 53 different plasmid with 53 different guide RNAs, including controls, and then pulled them together to make a virus. We also simultaneously generated the stem cell line, which expressed Cas9 already on the DOX-inducible promoter, and then transduced cell in day 0 and let them go. Knockout happened in 2 days, and then sorted them based on the BFP, and 2 different time points, the sequenced them into 2 different phases, like a single cell RNA sequencing and multi-omic. Okay, so what's multi-omic? Okay, before multi-omic, let's look at this. What is single cell RNA sequencing? It basically relies on each cell is uniquely, all the transcripts from the inside the cell is uniquely labeled with unique barcode, which we call the cell barcode, and it gives you about the transcriptome of the whole data, okay, from each cell. However, there are two key differences between multi-omic and single cell RNA sequencing. So multi-omic, along the labeling the messenger RNAs, multi-omic it also labels simultaneously accessible DNA regions from the nuclei. So it gives you transcriptome data and attack data as well from the same cell, which is kind of cool. Another key difference here is this is based on the whole cell. So you get the mRNA from the cytoplasm and nuclei. However, in multi-omic, you get mRNA from the only nuclei. That's the thing to keep in mind. So we did 2 different time point sequencing, and we got data back. And then we had a reasonable amount of cells, more than 10K cells in different time points, only like day 3. They had to do like filtering out the bad cells. However, if you look at the number of genes and the number of reads per data set, they're kind of the different time points, 2 different methods shows reasonable comparable distribution against each other. Okay, now we sequence the data, we have the sequences, now we have to identify which cell has got the which perturbation. So from, again, back to our construct, it's like a mRNA is generated from this BFP with a guide barcode at the end of it, and it's attached by cell barcode and then unique molecular identifier, which is internal part of this the pipeline. So we align our data to BFP-based custom genome and identify how many of cells has actually this transcript being the cassette being expressed. Actually, you can see most of the cells has this transcript being expressed as their BFP positive, but there are still some cells which has shown as a red are not expressed in BFP. Either they didn't get it or it got shut off like later time point. So next question is, okay, since we have a guide barcode now, how we can assign each cell their guide identity, right? So it's already sequenced, it should be in a sequencing data. I'm going to show some raw data here. So simply saying, if you look at your data, you should be able to capture sequences of guide barcode, which is already there, and each like guide barcode has been assigned unique cell barcode and UMI pairs. So simply you have to search for guide barcode and find that all cell barcodes, right? So we did that. We searched through the all guide barcode sequences, and I'm going to show you like how much of the cells we could recover. We could identify assign any guide barcode. As you can see, we couldn't recover all cells. We couldn't assign the all cells their guide barcodes just simply searching. The difference is even striking for the 11 time points. You can see only 30% of the cells could be identified. So it's telling us actually this standard pipeline, you cannot assign perturbation effect or guide identity to the different cells. So you have to do something else. So we came up with we followed up with the enrichment PCR step. So simply saying you are going back to your library, designing certain primers, which covers those guide barcode, cell barcode region. It's already like an established library. And you over amplify it with PCR and sequence again, is hoping to capture more cells with the guide barcode. But the important thing to keep in mind here, when you do PCR from the single cell library, it's easy to mess it up because you are amplifying and introducing more noise. You might be like introducing mismatching with different guide barcode with unrelated cell barcode. And then another key point here is each guide barcode should uniquely have unique cell barcode and UMI pairs. None of CBC UMI pairs can be assigned to different guide barcodes. That's the important thing here. So we did PCR, and then what I'm showing you here is all the CBC UMIs we could recover from the enrichment PCR, showing on the like x-axis, and then y-axis showing us how many different guide barcodes they have been assigned to. As you can see, 90% of the data has been assigned to single guide barcode, which shows data is mainly clean, and we of course filtered out those like has been assigned to multiple guide barcode to prevent the confusion. And another important thing when you do PCR like amplification, you have to normalize the copy number variation. So what I'm showing you here is how many times each CBC UMI like GBC CBC pair has been amplified. So you see some of them has been amplified like heavily, others are present in like multiple copies or 1 or 2 copies. So it might be saying there's like a like fake data like introduced into doing the PCR. So we applied the Gaussian mixture model, which is famous model to separate the true population from wrong population, simply saying we focused on these truly amplified populations. So let's see what data looks like. So after this dialogue PCR and cleaning, as you can see, we could increase the number of cells assigned to particular guide barcode. The difference is even striking for the 11 data. It went from the around 30s to around 80% and 90%. Okay, and it helped us. So looking at the basic stats, how many cells we get per guide barcode, the range is around like a 250 and 300. Also, most of the cells are single hit. What does it mean? It means most of the cell has got a single guide RNA, and we could expect that one gene to be knocked out, I think, which is important to study the knockout effect in individual level. Or like summing up after this cleaning, we got assigned 80% of our cells to different guide barcodes. All 53 of our regulators and then plus controls could be captured from data. I ended up having 250 and 300 cells per guide barcode, and then majority of the hits were single hit. All right. So now we have a data cleaned up and then separated. Now we can look at the analysis. So what I'm showing you here is a log-fold change in the mean expression ratio between the perturbed cells and control cells. So first gene here is our potential regulator, which is aimed to be knocked down by guide RNA. So idea here is that any other gene is a predicted targets of that gene. So if you see downregulation in your regulator, you should be expecting to see downregulation in your in their target as well. So in initial effect, it looks like there's a downregulation in most of its target, but it's like a small set of gene, and then looking at only like naively to like comparing the perturbed to unperturbed population. But we decided to do more in-depth analysis and looking at the global level of the perturbation. Therefore, we apply the dimensionality reduction clustering approach to identify the perturbation effect in a clustering pattern. I'm gonna tell you how. So here we are. What we are seeing is lower dimensional representation of data, colored with time point and then condition. You can see colored by perturbed and unperturbed. Simply perturbed means any cell that has been assigned to any guide barcode. So we know there's guide RNA there. And unperturbed cells means they haven't been assigned to any guide RNA, so we shouldn't expect any perturbation here. So as you can see by the color, so green and then which is day 3 perturbed, and then the orange, which are like a day 11 perturbed, they're kind of capturing the left side of the graph. However, like day 3 unperturbed and then day 11 unperturbed, which are the purple and yellow, they're kind of showing the different distributions. They're kind of separating. Unperturbed and perturbed data seems to be kind of separating from each other. These are just individual plots per condition per time point to show you how they capture the transcription space. So we first thought it might be actually affected by depths, right? So if you have a lower depths in unperturbed cells, they might show different occupation of transcription space. But when we compare the depths of the perturbed to unperturbed in different time points, actually we see there is no differences in the perturbed that can cause this differentiation. I mean, differential occupation in the transcription space. And most importantly, when we look at the marker genes, which are the markers of pluripotency, they're kind of more expressed in unperturbed cells, but less expressed this part of the graph, which are perturbed cells, which makes sense because you are looking at the major transcription factors. It might be shifting, they might be losing their transcription, their pluripotency state. So we decided next to apply a method called non-negative matrix factorization, which is used as a dimensionality reduction method. So simply saying I have a big matrix, it is a way of representing your big matrix as two smaller matrices. And then important thing here is the product of these two matrices should be closest approximation of your big matrix. So idea is to identify the important features. It actually happens to be like a major recommendation system in the Netflix as well. They use this to identify the features that you like in movies, and they recommend the movies based on that. But as we know, single cell data is more interesting than the Netflix. I'm going to show how it's applied to single cell data. So here we have a raw data, the n number of genes and n number of cells. They apply NMF. NMF finds the important features and it comes up with a lower dimensional two matrices, like a gene matrices and cell matrices. So we can apply clustering approach to the cell matrices based on those identified important features, then do clustering and identify the clustering pattern of our data. It is actually similar to principal component analysis, right? To identify important features and cluster your data based on this. But this method is more robust. Okay, what I'm showing you here, the 15K clustering based on the NMF identified features, and you can see how each different clusters kind of occupies different transcription space. And on this table, we are seeing the cluster assignment of, again, different time points and different conditions. So interestingly, you can see like a perturbed cells and unperturbed cells are kind of showing different cluster occupation. For example, in cluster 5 and 6, mainly it's like a day 3 perturbed, where there's no unperturbed. In day 7, and then cluster 7 and then cluster 10, it's mainly unperturbed day 3, but not perturbed. Difference is less in day 11, but you can still see the pattern. For example, in day 11, in the cluster 11, you can see mainly it's day 11 perturbed, where cluster 12 is unperturbed. So basically, they are unperturbed cell and perturbed cell, kind of showing the cluster present, differential cluster presence. Okay, so let's look at how our data actually perturbed populations are lying over. So simply saying, all these cells here has got the guide RNA which attacks the NR6A1 gene, and this in this plot we are seeing all the cells which has got the guide RNA which attacks the CTCF gene, and in this plot we are seeing all the cells which has got the non-targeting guide RNA which are controls. So as you can see, they are actually all perturbed and controls are occupying the all transcription space. So it's thought to be like due to noisy feature of the single RNA sequencing data. So it doesn't actually make much sense to compare whole perturbed cells to whole control cells. So people have been coming with idea to find the local, the perturbation effect, which basically means instead of comparing the all to all, you should compare perturbed cells in the same transcription to control in the same transcriptional space. But how would you define perturbation locality, right? So again, in the field, people have been using the different approach to that. We thought like a boundary, setting the boundary by NMF-based clustering, simply saying, look at the cluster, compare any cells, any perturbed cells to control cells within a cluster, and look at the differential expressed gene. In that case, gene 1 and G2 would be differentially expressed, and it would be network of CTCF, and we would say there's the edge between CTCF and gene 1 and then CTCF and gene 2. And do that for all clusters and all perturbed cells, and then finally come up with the perturbed network. Okay, now we have network, so what to do next? So of course you have to compare your network to existing networks. Okay, I promise this is my last slide, this fancy terminology. So there are certain metrics you have to use when you do compare networks, which are recall, precision, and F-score. Simply saying, recall is saying how much of the true network you can predict or you can recover. And precision is opposite. It is saying how much of your predicted network is actually true, which is same as specificity and sensitivity we use in the wet lab. And F-score is kind of harmonic mean of both, right? So what I'm going to show you here is a comparison of my identified perturbed network to Merlin network in different thresholds as like a blue and an orange bar, and also already known network from the ESCAPE database shown as a gray bar. The first thing I want to look at the gray bar because I can see like precision and recall is higher, and obviously F-score is higher too. So it means actually our identified perturbed SIG network has some degree of overlap with already published network, which means actually they're reproducing the same network, which makes sense. But when looking at the Merlin predicted network, we can see recall is smaller because, as I said, recall is saying how much of the known thing you can predict, because perturbed SIG network is so big, like 20,000 edges, where a Merlin top is like a few hundred, so you would expect recall to be lower. But the interesting thing is precision. So you can look at the precision in different thresholds of the Merlin. So it's simply telling us, if you look at the top predicted Merlin networks, 20% of this network has already been validated by perturbed network we have there. So again, saying this network is so big, and then you have to do like different threshold and comparison. But I'm going to show you like interesting key part of the network. So here we are seeing the OCT4, which is also known as a PO5F1, and SOX2 as a major regulator of the pluripotency. And it's already known there's an edge between them, right? So it's predicted by Merlin and then validated by perturbed, but all other downstream targets of these two genes wasn't in the established data. It's predicted by Merlin and then validated by perturbed. So it tells us actually perturbed might be like a reasonable approach to do high throughput validation. But there are still lots of things to do. For example, I didn't talk about the multiome at all. We need to apply the same pipeline to RNA-seq part of the multiome and then see if we can recover the same network components. And most importantly, we can use ATAC-seq data, which gives us accessibility, and we can look at the accessibility of the perturbed and unperturbed cells to see how they change and further refine our perturbation effect and final networks. And we can also apply network component analysis, which is dimensionality reduction for networks, to filter out the noise and then come up with a consistent network. So although this is like a reasonable approach, there are still like use for this approach. For example, we used single guide RNA for regulator, but you know, like efficiency might be low. So in the future considerations, it might be good to use more guide RNAs, like 4 or 6 per gene. And second problem is you cannot actually look at the mRNA level of the perturbed gene and say if it is perturbed or not. You know, as HARCAS9 works, right? It makes a double strand break. So although it might cause a knockout, but mRNA level might be still there. So you cannot look at the mRNA level and tell if it's worked or not. So as a solution, we can use CRISPR-I based, which is basically using the dead Cas9, which means catalytically inactive, and it cannot create a DNA break, but it can sit on the promoter or physically prevent the transcription happening, or it can have a repressor domain fused it and then bring the epigenetic markers and then close the accessibility of gene and then stops the transcription. Then you can look at the mRNA level and say, yeah, this gene is not transcribed. It might be knocked out. And then third most important thing, I think, as I told you, like enrichment PCR step, because since finding perturbation identity is relied on the indirect labeling of guide RNA with guide barcode, it is actually hard to recover. You have to go extra step of doing enrichment PCR. But new direct capture method allows you to directly capture a guide RNA expression. So basically, they have a specific sequence, which is also captured during the single RNA sequencing. So once you have like expression data set, it's going to give you information about the guide RNA directly as any other genes expressed in the cells. And speaking of rigor and reproducibility, we try as a computational and wet lab, we try to do certain steps. For example, we try the explicit documentation of the algorithms, and then analysis we do, and then record every step. I put a script and protocols, which is relevant to computational lab and the wet lab. So we try to, for genetic analysis, everyone tries to use the same script and the same protocols. And if we have something suspicious, like a results or something too good to be true, we ask other lab members to reproduce same data using our commands or scripts. And our algorithms and the documentations are available in the GitHub as well. You can access this QR code. And then not to mention, we do experimental validation of algorithms prediction, because my whole presentation was on the validation. I'm very grateful to have 2 PIs and 2 lab members, and very grateful to my committee for their support during my project. Yeah, any questions? I hope you find it interesting. Yes, please.
Why do you think that would be allowing to use mRNA levels that's a better readout? Wouldn't both of those prevent transcription?
So yeah, speaking of here Cas9, how Cas9 works, like it creates a double strand break in the DNA, and then DNA repair pathways comes, like non-homogeneous end joining, and it's likely going to introduce error, right? And then assume this error will cause the transcription to stop. But actually, this error doesn't stop transcription. So regardless of error, it might be still transcribed, and this error might be bringing the RNA degradation pathway to degrade the RNA later, or even might not be even degrading. So it might cause like a frame shift. Protein might not be there, but mRNA might still be there. So you might see have a knockout, but you might not see it in a mRNA level. But the idea of dead Cas9 is it is physically, or it basically blocks the accessibility of the polymerase, and then stop it from going and transcribing the gene. So if it is like taking an effect, it is certainly going to stop the mRNA production. But in Cas9 base, there's a possibility mRNA might still be there. I hope that was clear.
And the issue with the possibility of mRNA is still there is that it's still sequenced and counted.
Yes, you count as like a there. Exactly. So it gives a loss, the false negative. We are saying, oh, mRNA is still there. We can't say if it's knocked out or not, because we know like RNA sequencing is purely based on the mRNA levels. Yeah, thank you. Any more questions? Was it too confusing? Yes, please, Raj. Sure.
It's a very cool project.
Yeah.
I might have missed some of the details, but so you have a developmental trajectory, correct?
Yeah.
So you have expression at multiple time points along the development, right? Is that correct?
So in this case, you mean the expression of the transcription factors we are targeting?
Yeah, and then the perturbs.
Yeah. So in this case, we have we call it time pointed data. Maybe I should go fast too. Here, yeah. So we basically we have a stem cell growing in the culture, and they maintain their status, and we have a transcription factors that potentially might be helping to maintain their status and might not be known. And we actually sequence them in 2 different time points. It's not because of actually there's a time series or developmental thing. We expected to see the same effect, but we wanted to include the later time point. One, because of as I explained to Chris earlier, you might be knocking out the gene, mRNA might be still there in early time point. We thought maybe we should give it more time if gene is truly knocked out. So it should all mRNAs hanging out there should be degraded. It might give us a clear and more clean data. So I give it.
The cells are differentiating, the ones that don't contain the pluripotent state.
So they should, yeah, they should differentiate.
I guess I'm if I understood, so you have perturbed and you have unperturbed cells. And sorry, are the unperturbed cells, they get the Cas9 or whatever you're using, antivirus.
Exactly.
Or you just didn't sequence it then.
So we sequenced the cells, but we couldn't capture the guide RNA in them. We call them unperturbed cells.
And why is, but what, are they really, are they intended to be unperturbed?
That's a great question. That's a great question. So they are not intended to be unperturbed cells. We included control non-targeting guide RNAs, and sorry, yeah, here we included control guide RNAs. The idea was this. So every cell we get the guide RNA, I will find the cells that has a guide RNA. I will also find the cells which has a non-targeting guide RNA, which will be my control. I will be using that. But we find out actually some of the portion of the cells doesn't even express any, neither like a guide RNA or non-targeting. We call them unperturbed. And the important key here is we didn't use unperturbed cells while doing network comparison. We used only those cells which we can recover the guide non-targeting guide RNA. So we know construct is there, they are expressing non-targeting guide RNA, and we only compare perturbed cells, control cells, not the unperturbed cells. I include them in the visualization to see what's happening with all those unperturbed cells and how do they behave. So in final networks, they are not included in the comparison.
Right. That makes sense.
Yeah.
So now, the last question is,
Yeah.
so you are trying to, I think you mentioned, you are looking, you're comparing, to get your network, you're comparing the perturbed cells and the control cells from the same region of the human, is that right?
Yes.
But what if, but that's confusing to me, but so, again, misunderstanding the model, aren't the control cells pluripotent? They're maintaining a pluripotent state. And are the perturbed cells differentiating? Why would they be in the same part of the human? I guess that's what I'm thinking.
So yeah, in ideal world, you would expect perturbed cells being in the one part of the graph and then control cells in other part of the graph if you had a clean, no noise data. But we know like there are lots of, because of the dropout effect and then technical effects, we have lots of like noise in the data set, right? So people have been trying to clean up this and then coming way of like identifying this. So the idea of comparing the within cluster perturbed to control is this. So if they are occupying the same region, we assume they're affected by noise in the same way. So if I compare this perturbed cells to control in other parts of the graph, it means they are actually occupying the different part of the graph because there are lots of noise there and they are not in the same transcription space. But the closer, like a closest way of doing it, like better way of doing the thing is like to just look at the cluster and then compare the control to perturb within a cluster, because that will tell us they are affected from the noise and technical issues like in a less way compared to other way. I hope that explains the reason.
You might be expressing the guiding need, but it's not.
It's totally possible. It's totally possible. So you might still be guide RNA there, and then it might not be working. That's another shortcoming of this method. That's what I was saying. That's why we should replace this with the CRISPR-I to see network there. And then there's like a more, it would be more profound way of showing knockout is happening or not. So that's totally possible. That's why we are doing all this clustering and then different competition approaches to try identify truly perturbed cells. Any more questions? Yeah, Chris, please.
The reading this noise direction compare locally similar populations of cells that I identified as control perturbed and is that what the non-native expectation was? Or like, what do you, how do you generate those clusters?
So non-negative matrix factorization first tells us what are the important features that defines your data. It's basically a feature selection method. Identifies the, we have like a 20,000 genes there, right? It identifies that what are the major genes that actually cause the differences between all your cells. So it tells us what are those factors. Then we apply clustering to those identified factors. We don't apply the own yield space, but the only like NMF, the factors NMF thinks is important, and then do the clustering. In that way, we just try to identify internal pattern of the data. That makes sense.
So you're picking out gene features based on.
Yes, important gene features. Exactly. Finding the most important genes that are, causing the difference in the data.
Wouldn't that increase your noise in this case? If you're worried about a lot of background noise, then you're probably picking the highest variable genes. The highest variable genes of noise, that's probably actually increasing the noise that you're getting from clustering.
So dimensionality reduction and feature selection is like a standard approach in the RNA-seq analysis method, right? So you cannot just look at the whole gene space and then cluster data, because you will be like clustering noise basically.
Yeah.
So you have to find what are the different features there. And then there are different approaches to do that. Like, as I mentioned, principal component analysis is one of them. But the network, actually, an NMF approach is supposed to be more robust and then more better than the principal component analysis, is giving you more important features. That's why we picked the NMF to continue this.
But in the end, at the end of this, you clustered on the NMF features. You're still comparing within cluster between control, targeted, and target.
Yeah. So I understand what you're saying, trying to say like even even after clustering, it might be affected by the noises. But one way of overcome this to do like a different way of clustering, like how many clusters would you choose? That's another question to ask here. So we did the range of clustering. So you don't want to like under cluster. You don't want to have like most of your data in one cluster, and you also don't want to like over cluster. You don't want to have like a few cells in like a one cluster. So we did a range of clustering, and then used all the consensus of comparison between all different clustering, and then come up with a union. So we are considering different clustering options, possibly like filtering out those noise, which is like entrapped in the cluster, as you said.
Did you previously use like clusters into like pathway analysis to see if there's any sort of enrichment in any of these clusters as like a sort of validation step?
Yes, we did actually. I didn't show it here, but we did that step 2 to see which cluster are more enriched with perturbed data and then more enriched with control data. So it is another way of considering which cluster to take into account, looking at the whole cluster or look at those clusters which has enriched. That's definitely the approach we are using as well.
Enrichment of the gene. So you can get which cell cluster goes, which gene cluster, so we get the enrichment of genes in gene biology cluster. You can see from the level of the gene The problem with the mindset is that the populations are very subtle. It's a very slight population. It's like not like big. And we were hoping to see that the control cell is in one part of the state and the product cell is in the other one, but that doesn't happen. And that's not only our data, it happens for all product data. And it's because what the man said, there's too much noise. And then in cluster, you're kind of projecting the cells in the same systematic bias. And that third system is absolutely. But yeah, product data is noise, that's what we want to do.
Good. Thank you so much.